hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So here we are in biomolecules part 3. So I hope you're enjoying this chapter of biomolecules. So we are understanding what is exactly inside a tissue, how are these compounds formed, what are the bonds behind them, what is their structure, what is their composition. So that is what we are discussing in this chapter of biomolecules. So today it is lecture 3. So in the first lecture we found out the evidence that yes, we have organic molecules, we also have inorganic molecules, and we went on to talk about what is an amino acid. So in the second lecture we continue to talk about what is a lipid, which is comprised of fatty acids and glycerol. Then we went about to talk on your nucleotides. So what are the components of your nucleotides? Then we spoke about your primary and your secondary metabolites. So today we'll be moving forward from there. So we'll talk about the structure of biomacromolecules, that is your proteins and your polysaccharides. So that is the agenda for today's class. All right, ready? Shall we move forward? Okay, so here we go. Just give me a minute. So are you able to recollect whatever we read in previous class? So here we go. So giving you a quick recap. So we started talking from lipids. So before I start with lipids, let me write the experiment. So you take a living tissue. You homogenize it with your trichloroacetic acid. So then you filter it. So you have two fractions. One is your filtrate, or your acid soluble portion. The other one is your retentate, or the acid insoluble portion. So what is present in your acid soluble portion are your biomolecules, or we can also call them as biomicromolecules. Right, so they have weight of around 800 Daltons. At the maximum it should be 800 Daltons. So here they are biomacromolecules. So they have greater than 800 to 1000 Daltons. So who comes under this biomolecule portion and who comes under the biomacromolecular portion? So biomolecules, so you have your amino acids and you have your lipids, then your nucleotides. So here biomacromolecules, you have complex molecules like your proteins, your polysaccharides and your nucleic acids. Right, so here I have star marked lipids because lipids are actually biomicromolecules, but they come in the macromolecular portion because they cannot readily dissolve and come out. Right. So that is the reason it is always it is actually conjugated with your other components like your phospholipids, glycolipids, etc. So that is why it is present in your biomacromolecular portion, but it actually belongs to your biomolecules. Alright, clear? Okay, so then we went on to talk about your lipids. So if you remember, we spoke of the composition of lipids to be fatty acids and glycerol. Right? So fatty acids can be 3 molecules of fatty acids and you have one molecule of glycerol. So fatty acids are organic molecules with the R group. So you can be triglyceride, diglyceride, or monoglyceride, right? So we spoke about saturated fatty acid and unsaturated fatty acid. So saturated fatty acid has no carbon-carbon double bond. So there is no carbon-carbon double bond here. So here there is a carbon-carbon double bond. So example of your saturated fatty acid will be your palmitic acid. And your unsaturated fatty acid can be your stearic acid. Right? So moving further, you have something called as polyunsaturated fatty acid. So polyunsaturated fatty acid means they have more than one carbon-carbon double bond. So they have more than one carbon-carbon double bond. So that is your polyunsaturated lipid. So example of this is you have your arachidonic acid, your oleic acid, linoleic acid, linolenic acid, etc. So which is essential in your body? That is your unsaturated fatty acid. That is essential because it cannot be synthesized in your body. It is essential because body cannot synthesize them. Right? So they have to be supplied through the diet. At the same time, these are very reactive. Thus, they get easily metabolized. And they are always liquid at room temperature. Okay, fine. So your saturated fatty acid is non-essential. They are solids at room temperature and they are non-reactive. So they cannot easily get metabolized and they get stored in your body as cholesterol, etc., thus causing obesity. So this is what we read about fats and about lipids. Then we went on to talk a bit about your nucleotides, if you remember. So talking about nucleotides, so your nucleic acids are made up of polymers of nucleotides. So your nucleic acids, they are made of polymers of nucleotides. Okay. Hi W. Yes, a very happy good afternoon. So I hope you're doing well. So thank you so much for joining my lecture this evening. So please do like, share and subscribe. So nucleic acids can be your DNA and your RNA. So nucleotides are made up of two components. First is your nucleoside. The second one is your phosphate group. Right? Your nucleoside is further made up of a sugar and a nitrogenous base. So sugar can be two types of sugar. One is deoxyribose sugar. The other one is simply ribose sugar. So deoxyribose sugar is in case of DNA and ribose sugar is in case of RNA. Okay, so nitrogenous bases are further two types. One is your purine and the other one is your pyrimidines. So purines are double membrane ring structures like this, right? So who are the purines? So you have two purines, that is adenine and guanine. Your pyrimidines are single ring structures like this, and you have 3 pyrimidines. The first one is your cytosine. Then you have uracil and thymine. So uracil is specific for your RNA, thymine is specific for your DNA, but cytosine is common to both RNA as well as DNA. Always remember, in your DNA, A always pairs with T. A is adenine, T is thymine, okay, in your DNA. And your G always pairs with C. So this bond in between is your hydrogen bond, okay. So this is in case of your DNA, right? So in case of your RNA, so your A always pairs with U and your G as usual pairs with C. So please remember this, okay. This much is easy. So now we move further. Suppose I write I have a nucleoside here. Let's say I have a nitrogenous base here, okay. Then I have a sugar here. So now I should have a nucleoside. Adding phosphate to it, I should have a nucleotide. So let's form the molecules. So the first molecule let's take is adenine. So adenine, if it binds with deoxy sugar or simply ribose sugar, what will be the molecule? So if it binds with ribose, it is deoxyadenosine. So this is adenosine simply. So imagine it is getting added with the help of a phosphate group. Now it is forming a nucleotide. So what will be the nucleotide here? So here the nucleotide will be deoxyadenylenic acid. So here it will be simply adenylenic acid. Clear? So now you should know adenosine is a nucleoside and adenylenic acid is a nucleotide. This is very, very important. Alright. So now let's write for guanine. Deoxyribose sugar and ribose sugars. That is, I am writing for DNA as well as RNA. Okay. So deoxyribose sugar, it has deoxyguanosine. And here I have guanosine. Now when I combine it with the phosphate group, so what do we get? We get a guanylic acid. Okay. So this is so here it is deoxyguanylic acid. So here it is simply guanylic acid. Understood? So now you know which is a nucleoside and which is a nucleotide. So now let's go to thymine. So mind you, thymine cannot have two sugars because thymine is present only in your DNA, right? So I only have the deoxy sugar. So it is only thymidylene. So it is thymidine. So with the phosphate, it forms thymidyllic acid. It forms a thymidyllic acid. Alright. So now we go for uracil. So uracil binds with only RNA, so it is ribose sugar, so it is uridine. So plus your phosphate group. It is uridylic acid. Fine. So now the last one is cytosine. You combine that with your deoxy sugar. Because cytosine is common, I write ribose sugar as well. Okay. So it is your deoxycytosine. Sorry, it is cytidine, not cytosine. It is cytidine. And here it is simply cytidine. So when you add a phosphate group here as well as a phosphate group here, so it forms deoxycytidylic acid. Here it is simply cytidylic acid. Fine. So now I want you to remember which is a nucleoside and which is a nucleotide. So that is the main reason I wrote the entire thing on the board. Clear? So you should know which one is a nucleoside. So these are all nucleosides. And here you have your nucleotides. So make sure you never forget this. Alright. So shall we move forward? So here we move. So we spoke about your metabolites before that. So primary and your secondary metabolites, right? For example, when you eat your food, the products that you get are your primary metabolites, that is your glucose, your amino acids, fats, etc. When that is used inside a process, so you get further metabolites, which are your commercial products, that is your secondary metabolite. Okay. So common to all those compounds, so biomacromolecules are present in your acid soluble pool, right? So common to all the compounds were present in your acid soluble pool. So acid soluble pool is your filtrate, right? So the molecular weight of your biomicromolecules of your acid soluble pool was only 18 to 800 Daltons. So here this is your biomicromolecules, or simply your biomolecules, right? But in the biomacromolecules, you have 4 types. So you have proteins, nucleic acids, polysaccharides and lipids in the acid insoluble portion, right? So these are biomacromolecules. So lipids are strictly not macromolecules, they are strictly biomolecules. But since they are conjugated with each other, they come in the macromolecular fashion. Alright, so I hope you remember metabolites now. First, metabolites are end products of metabolism. Alright, so the end products of physiological process, if there's a physiological process, say for example digestion, so the end products of digestion are amino acids. Say for example you have sugar, so they are called as your primary metabolites. Okay, so when these enter inside a metabolic pathway, so they produce commercial products like your alkaloids, your drugs, your pigments. So those are called as your secondary metabolites. Right? So then we have your biomacromolecules. So biomacromolecules have your proteins, you have your polysaccharides, and you have your nucleic acids, right? So proteins are polymers of amino acids. So we will talk much in detail about the proteins. So proteins are polypeptides. Poly is many, right? And peptides means it is made up of peptide bonds. So it is made up of linear chain of amino acids which is linked by peptide bonds. So here if I have one amino acid, so I have the second amino acid, I have the third amino acid, and so on, right? And proteins are heteropolymers, meaning it is made up of different amino acids. Not all amino acid will be the same. It is made up of different amino acid. So that is why your protein is a heteropolymer. So it is a polypeptide made of your amino acids, and the amino acids are very different. So it is heteropolymer. So it is a linear chain. Yes ma'am, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So dietary proteins, so we know what are essential proteins, that is their source of essential amino acid. So what is the important function of these proteins? They help in the transport of nutrients across the cell membrane. If you remember facilitated diffusion, you have carrier proteins, right? So they help in the transport. Then they help fight infectious diseases like your antibodies. Your antibodies are also proteins. Then they have some hormones. So you have your insulin. Your insulin is a hormone, and some are enzymes. Mostly 95% of your enzymes are proteins, right? So collagen is the most abundant animal protein that is present, and the most abundant plant protein is your ribosome. So that is your ribulose, bisphosphate, carboxylase, oxygenase, and it is the most abundant protein on the biosphere. Right? So this is a tabular column that's provided in your textbook, and I want you to look into this tabular column. It's very important because in last year NEET this came in the form of a match the following. So this is very important. So collagen, intercellular ground substance. We have read this in your connective tissue and structural organization in animals. So trypsin is a type of enzyme, insulin is a type of hormone, antibody fights infectious agents, receptors, sensory receptors like your if I can taste something, so I have my someone has to receive my taste and take it to the brain. Those are the receptors. Then you have GLUT flow. GLUT is your glucose transport. You have 1 to 4. So this is the fourth one they're giving you as an example. Alright, so we will stop here. We'll talk much in detail about the structure of protein and polysaccharide on Monday. So until then, have a very happy weekend. So please go through the NCERT portion. If you guys have any doubt, get back to me. Alright, so thank you so much for watching my lecture, and please don't forget to like, share and subscribe. So until Monday, it's me signing off. Thank you and bye.